Mutations, chromosomal abnormalities, and genetic disorders. A mutation is a permanent change in DNA's sequence of bases. If the change in DNA is within a gene, it can be copied into RNA during transcription and alter the protein. There are many types of mutations that can alter a gene expression and result in an altered protein synthesis. We will cover point mutations, insertions, and deletions. Point mutations, or substitutions. What would happen if one base was changed? Let's use a sentence analogy by changing the C in cat to a B. The correct sequence is the cat ate the rat. Point mutation might look like the bat ate the rat. The meaning of the sentence is changed. The same type of mutation can happen in DNA with varying effect. Insertions. What would happen if one base was inserted into a DNA sequence? Continuing the sentence analogy, add a B before cat. The correct sequence, the cat ate the rat. An insertion mutation might look like this. And then there's a B inserted here. So now the rest of the sentence looks like this. This new sentence makes no sense. Deletions. What would happen if one base was deleted from a DNA sequence? Delete the C in cat to represent the deletion of a base. The correct sequence, the cat ate the rat. Deletion mutation, the ATA. So see, now the C is missing. Again, the new sentence makes no sense. Insertions and deletions can be particularly dangerous mutations, since it is more likely that the meaning of the gene will be damaged. We learned earlier about mutations that can occur in DNA. Since the change is in the DNA, the mutation is inherited by later generations. The mutation of a normal allele produces a mutated allele for that gene. Inherited disorders are usually due to recessive mutated alleles. In general, mutated alleles can have no effect, providing neither an advantage nor disadvantage, be harmful, in which case they may be reproduced at a lower rate and fade away in the population, or be helpful, in which case they may be reproduced at a higher rate and become prevalent in the population. In some cases, having one mutated allele may offer an advantage to an individual, while having two may be harmful. This is related to the recessive nature of mutated alleles that persist in a population. If there is an advantage to individuals who have one mutated allele and the percentage of the population with just one mutated allele is high, then this may provide an overall advantage to the population. Those with no mutated allele are unaffected. However, those with two mutated alleles may inherit a disorder. A mutated version of the hemoglobin gene is responsible for the sickled hemoglobin protein. Having one gene with this mutation provides protection against malaria. In places where malaria is a leading cause of death, this mutation is an advantage. However, having both genes with this mutation results in sickle cell anemia, which seriously harms individuals. Where malaria is a serious threat, this mutation seems to have a net advantage, so it persists. Click for a video. What is malaria? People with sickle cell anemia carry a different allele of the gene for hemoglobin. The sickled allele is due to a point mutation of the normal allele. Here we have the mutation, there's a T instead of an A. What happens to the hemoglobin protein when this mutation is present? Use the codon wheel to determine the amino acids. Alright, so CUG. We start at the center. C, then U, then G gives us LEU. ACU gives us THR. CCU gives us PRO. GAG gives us GLU. This is the same. GAG will also give us GLU. Then AAG gives us LYS. And there we have all the normal hemoglobin amino acids. Here we have the sickled hemoglobin RNA. This one has changed, so all the rest will be the same, but let's just go through it again. CUG, CUG gives us LEU. ACU gives us THR. CCU gives us PRO. Now we have GUG. GUG gives us VAL. GAG gives us GLU, and then AAG gives us LYS. So this one has changed. Here we have the sickled hemoglobin amino acids. The RNA transcribed from the sickled allele produces an A instead of a U. The protein is built with Val instead of glue. The shape of the protein is changed because of the changed amino acid, and the shape of the protein determines its function. With one sickled allele, the individual is resistant to malaria. With two sickled alleles, the individual develops sickle cell anemia. The hemoglobin will not deliver oxygen to the cells efficiently, and the individual is very susceptible to malaria. Malaria is currently present in 100 countries and threatens the lives of about half the world's population. In areas where it is a serious threat, the sickled allele persists for good reason. When only one allele is present, it confers a survival advantage. However, when both alleles are present in an individual, it leads to sickle cell anemia. As populations move away from regions threatened by malaria, it would be expected that the sickled allele would fade away, as it would no longer have an advantage when occurring by itself, but would still have a disadvantage when occurring in pairs. Here are other inherited disorders. 2 mutated alleles results in the disorder, while just one results in resistance to the indicated disease. Phenylketonuria is a buildup of an amino acid causing brain damage, but one mutated allele offers resistance to fungal infections causing miscarriages. Tay-Sachs is a buildup of a protein which causes nerve damage, but one mutated allele offers resistance to tuberculosis. Cystic fibrosis is when the body produces thick and sticky mucus that clogs the lungs, but one mutated allele offers resistance to cholera. And glucose 6-phosphate dehydrogenase deficiency is a missing enzyme that helps protect red blood cells from damage, but one mutated allele offers resistance to malaria.